This lab is your Unity and Diversity of Respiration lab, which you can find on pages 61 through 64 in your lab workbook. We're going to be starting over on 61 with the background information. So the question we are trying to answer today is, do all living things give off carbon dioxide? Background. We know that we get our energy from food through some sort of chemical reaction in our body. Since figuring this out, we've returned to the idea that we might actually be burning our calories. Burning involves the reaction between a fuel source of carbons, hydrogens, and oxygens, and the oxygen readily available in our atmosphere. The products are carbon dioxide and water and a bunch of energy. Since we suspect this may be similar to what happens in our bodies, in this investigation, we are trying to figure out if all organisms produce carbon dioxide, a testable product of the chemical reaction of burning. We already know of one way to test carbon dioxide, the indicator BTB, or bromothymol blue. Here, we're using a similar indicator called phenyl red. Yeast, a unicellular, which means one cell, microorganism, goes through either sexual or asexual reproduction. Yeast can grow in many different conditions as long as they have a food source, usually sucrose or sugar, for energy. They produce and release digestive proteins, or enzymes, into their environment. We place different kinds of organisms in test tubes with phenyl red to see if they cause it to change, which would indicate the production of CO2, or carbon dioxide. In Part A, you will run controls in known substances, helping you to properly interpret any changes you observe in Part B. You will use the results of both parts to try and answer the title question. Do all actively living things give off carbon dioxide? The materials you're going to see in this lab are the phenyl red, carbonated water, CO2 water, droppers, straws, paper towels, test tube racks, sprouted radish seeds, small test tubes with stoppers. There's actually aluminum foil for the stopper, brass screws, a yeast sugar solution, a boiled yeast sugar solution, dry radish seeds, and one small live insect. And what we have is a cricket. So right now, I would like you to pause the video and please go answer pre-lab questions 1, 2, 3, and 4 on pages 61 and 62. Procedure, Part A, characterizing the response of phenyl red. In the following series of tests, you will determine the response of phenyl red to carbon dioxide so that you can interpret the results of Part B. Set up 3 standard size test tubes in a rack and place a paper towel under the rack on which you write the numbers under each test tube, 1, 2, 3. Perform some tests with known substances to determine what color change a phenyl red indicates. Number 1, place phenyl red in your 6 small test tubes. Add the screw. Add different items to each tube. Place stoppers in the tubes. Observe and record the color change in the phenyl red over time. In each tube, place 10 to 12 drops of phenyl red. Then add the following. Tube number 1, nothing, phenyl red only. Tube number 2, 5 to 10 drops of carbonated water. Tube number 3, your breath. Blow gently through a straw for 30 seconds into the phenyl red solution. Now we're going on to part B on pages 62 to 63. The procedure that you need to read is on the bottom of page 62. And here it tells you what is in each of the tubes. And as the tubes get filled during this video, that will be repeated for you. Just so you know what some of these things are, yeast, as we talked about in the beginning, is single cell microorganism. We have radish seeds. These are dry radish seeds, which means they're dormant. They're nothing happens to them unless they get water added to them in order to start the sprouting process. Here you can see the sprouted radish seeds. You can see the tiny little baby plant or the embryo coming out of the seeds. Here we have a yeast and sugar solution that is not boiled. You can see it's like a whitish color. It smells like bread. And then we also took some of the yeast and we boiled it to a real high temperature. You can see the bubbles in there as it is boiling. These are the test tubes from part B, where you are comparing different organisms and the reactions that the phenyl red had when in the presence of these organisms. So here you can see set up tubes 4 through 10, right? This is right initially after they got set up, so you can see what the initial colors are. And then it was run on a time lapse. And this is a time lapse right here. I just gave you a short piece of it. And at the end of that time lapse, you will see the results of each of the tubes. So as you can see, these are the end results. And I'm going to show you each of them individually as well, or in groupings, I should say. So you can see here's 4, here's 5, and here's 6. Now here's 7, 8, and 9, and 10. So here they are a little bit closer. So you can see 4, 5, and 6, the colors. Here's 6, 4, 7, and 8, excuse me. And you can see them closer. This is 4, 7, and 8 in order. And then you have 4, 9, and 10, also in order. So you should be recording these results in your data table. Because it was a time lapse, you didn't catch the time for how long it took for the tubes to change color. So here's the data for that. Tube 6 is 20 seconds, tube 7 is 3.25 seconds, and tube 9 was over 24 hours, and this isn't shown in the video. Due to the fact that there was not data available to you for time because it was time-lapse video, this is the color change data. For tube 6, it was 20 seconds. For tube 7, it was 3.25 seconds. And for tube 9, it was over 24 hours. This is not shown on the video because of when the data was recorded. So you want to use this data to fill in the time for how long for a color change on page 63. Then you're going to go over to page 64 and you're going to answer the analysis questions 1 through 6 using your data from your data table on 63. And additionally, you are going to write a CER at the bottom to answer the lab question of do all living things give off carbon dioxide? And you're going to use your data table for evidence. How to write your reasoning is on page 2 of your workbook. And the overall rubric is on page 3.